So I'm going to look at two questions. The first question is, does the same virus amplified in two different media come out the same? And specifically, I'm looking at influenza virus amplified in either tissue culture or in air gallantoic fluid. The second question is whether the hemagglutination assay is an optimal method for quantifying influenza viruses used in serological assays. The HA assay has been used for decades, but now there are other technologies available, and I'll be comparing the HA assay to virus particle counting. A little bit of background first. There are several different types of influenza viruses. There are influenza B and C viruses that are found in humans. There are influenza A viruses of many subtypes that are found in different avian and mammalian reservoirs. The influenza viruses are segmented viruses. So this means if a host is infected with two different types of influenza virus, segments from one virus could be incorporated into another virus during replication of those viruses. And this process is known as antigenic shift. There are other ways the virus can change in antigenicity, and that's through mutations in the surface proteins, particularly the hemagglutination protein. And because of these changes in antigenicity, there is a need to continually monitor the different strains of viruses that are circulating around the world and periodically update the vaccine seed strains that are used in the seasonal influenza vaccines. So one of the pieces of information that go into vaccine seed strain selection is the hemagglutination inhibition data. And this data is generated from sera from people who are vaccinated with the previous season's vaccine, and then testing their antibody response to the currently circulating viruses. And to normalize everything, the currently circulating viruses are all titrated with the hemagglutination assay. And so this creates a level playing field when you're measuring the amount of antibody. After an incubation step, red blood cells are added, and then the plates are read to give you a hemagglutination inhibition titer. So to sort of understand this, we need to know what agglutination is. And if you take a dilution of red blood cells, leave them in a 96-well plate for a period of time, then they will pellet. If you take those same cells and add virus, influenza virus, it was discovered by G.K. Hurst in the 1940s that the cells agglutinated. That is, they formed a sheet and didn't settle down. And this was developed as a way of measuring the amount of virus. The amount of virus is normally, the virus is normally titrated in a twofold dilution. And you can see here with the filled circles that this would represent a 1 in 2, a 1 in 4, or a 1 in 8 dilution. And while it's still agglutinating, we take the HA titer as the last dilution step. So the HA titer shown here would be 8. Shortly after the HA assay was developed, people realized you could use it for measuring antibody response. So a 96-well plate is set up with a fixed amount of virus in each well, and that virus is incubated with a serial dilution of the antibodies. And if there's enough antibody to sequester the virus, it will prevent agglutination, and the red blood cells will pellet and precipitate at the bottom. So that's all fairly straightforward, but there are problems with this assay. One of the problems is that the different viruses have different surface proteins. Although it's all called hemagglutination, they vary. The influenza viruses isolated from humans tend to bind receptors that are alpha-2,6 sialylated proteins, whereas influenza viruses from avian species tend to bind alpha-2,3 sialylated proteins. Even even viruses that bind the same type of sialylated proteins can also bind with different affinities. In addition, it's been shown recently that the neuraminidase can affect the binding of the virus to the cell. And that has been shown for H3N2 viruses in particular. Finally, the red blood cells themselves can affect the assay. The different types of red blood cells have different types of sialylated proteins. For instance, red blood cells from avian species like turkey or chicken have more of the alpha-2,6 sialylated proteins, whereas red blood cells from guinea pig or type O human cells have more of the alpha-2,6 sialylated proteins. So as an example of how this can play out, I'm showing titration data from human influenza viruses that we used in the recent WHO serology to help determine what the immune response was to currently circulating viruses. We took several viruses that were grown in different media, eggs or tissue culture, and titrated them with different blood cells. The B and H1N1 viruses are all titrated with turkey red blood cells to a final titer of 8. The H3N2 viruses we titrate with guinea pig red blood cells, and this is because in the 1990s the H3N2 viruses lost their ability to agglutinate chicken red blood cells. If we take these same samples of virus and titrate the H3N2 viruses with turkey red blood cells, we see a difference in viral titer. This depends a little bit on the cells, on the media that was used to propagate the viruses. The cell-propagated H3N2 viruses didn't change in titer, whereas the H3N2 viruses that were propagated in air gallantoic fluid had a different titer. So obviously this is important for your assessment of a hemagglutination inhibition assay. If you're adding more virus, then you'll need more antibodies to sequester that virus to get the comparable HI titer. So HA, the hemagglutination assay, is not the only way of measuring virus. There are several other ways, and this is just a table showing a sampling of those methods. At the bottom of the table, you can see that I've mentioned things like plaque assay or TCID50. These types of assays measure infectivity, which is useful, but they take a period of time. They're slow and labour intensive. There are other methods like the single radial diffusion or ELISA assays. These can quantify viruses accurately, but they also require specific reagents to specific viruses. So of the assays that are very fast and are able to quantify viruses without any virus-specific reagents, the HA assay is at the top there, and flow cytometry is up there now. And there have been recent developments in flow cytometry that enable the detection and quantification of virus particles. And the data I'm going to present today was obtained using a virus counter developed by Endeavour and marketed by VirusSite. And they have a booth down in the exhibitors hall below. So the way the virus counter works is a little bit different from your regular flow cytometer. It's been specifically optimized to measure virus particles. That means the diameter of the that's flowing through the sheath fluid by the detectors is much smaller. So you're not measuring whole cells that would clog it up. It only measures small particles. In addition, rather than using specific labels, there is a combination dye which has two fluorescent, is comprised of two fluorescent dyes. One of the dyes binds proteins, the other dye binds nucleic acids. After an incubation step, the sample is run through the machine, and if the detectors detect the signal from both dyes, the nucleic acid signal and the protein signal, simultaneously, then it is registered as virus particle. If there is signal in one channel, it is registered as background. So those same viruses that I showed before that were all titrated to a final HA titer of 8 were measured with the virus counter. And the first thing to notice is that there is quite a range. This data is represented as particles per mil. So on the left-hand side, there is a log scale, and you can see that the particle counts per mil range from the mid 10 to the 6, right up to the high 10 to the 8. What was interesting for us is when we separated out the counts for the virus that were grown in tissue culture and compared those to the viruses grown in allantoic fluid. Shown with the blue diamonds as the average and the line as the range, you can see that the egg-propagated virus had less particles than the cell-propagated virus. And this was true for the B viruses, the H3N2 viruses, and the H1N1 viruses. And also of note, if you look at the values for the B viruses, they were consistently lower than the titers we got for the A viruses, even when the same red blood cells were used. So if you'll recall, the B virus and the H1N viruses were titered with the turkey red blood cells. So we wanted to investigate this further. We didn't know if this data was because all the viruses were different or if it was because of the media that they were grown in. So we used paired virus samples. That is, the same virus isolated and amplified in either egg allantoic fluid or in cell culture. We compared those, and I measured HA titers, looked at virus particle counts, and also the plaque forming units. So the first question I wanted to ask was, is there a correlation between the HA titer and the particle counts? And I also wanted to confirm that the difference between the egg and the cell cultured samples was real. So shown here is the B Massachusetts O2 virus with the virus particle counts on the left and the HA titer on the bottom. And you can see that there is a linear relationship between the particle counts and the HA titer. I wanted to push the system a little bit. So rather than doing a 1 and 2 serial dilution for the HA titer as I described before, I did a 3 to 2 dilution. That way I could capture samples that were at the upper and lower end of the HA. And so you can see where there are 2 bars for the HA. They were both of those samples were the same HA titer, but a different dilution. So even with those steps in the HA titer, there was a linear relationship. This data also confirmed that, as shown in the red, the cell-cultured samples had about 1 log more viral particles at the same HA titer as the egg-propagated samples. This data was replicated with the H3N2 virus and with the H1N1 viruses. So the next question I wanted to look at was how does this affect infectivity? So if we look at the number of particles and the number of plaques, we can get an idea of the population of the virus that is non-infectious. And again, I wanted to look at any differences between the egg and the cell-propagated viruses. So as you can see from this data, looking at the virus particle to plaque ratio, we got more viral particles than infectious plaques. And this has been shown for influenza viruses. Like others have shown before, we also got more non-infectious particles for the influenza A viruses than we did for the influenza B viruses. But what we also saw with these paired virus samples was that the number of non-infectious particles from the cell culture was about a log higher than the non-infectious particles from the egg-propagated samples. So just to summarise those pieces of data there is about 1 log more viral particles from the cell-propagated samples than with the same HA titer as the egg-propagated samples. There were more influenza A viruses than influenza B viruses at the same HA titer, and more non-infectious particles from the cell culture. So what does this mean? When we look back at those first 2 questions I asked, does the same virus amplified in different media come out the same? No. We have less particles for the same HA titer for viruses propagated in eggs. But there's also different binding affinity of those viruses. And so while we know there's a difference in the number of viruses, we're still investigating how that affects the binding. We don't know if we are looking at differences in packaging, if we're looking at differences in the HA protein itself and its ability to bind, or differences in the proportion of HA and NA on the virus, on the surface of the virus. So the other question, is HA the optimal method for quantifying influenza viruses in serological assays? This is still difficult to answer. When we looked at the data, we see a good correlation between the particle counts and the HA titer. But of course, you can change the type of blood cells you're using and change the HA titer. And also, we got that difference between egg-propagated and cell-propagated viruses. So part of the problem is that when you're using the hemagglutination inhibition assay, you're looking to measure the number of antibodies and the number of antibodies that are binding to viruses. So when you're trying to normalize the system, ideally you'd have the same number of virus particles. But for this assay, you also need to have enough virus particles that will agglutinate the red blood cells. And so that relationship depends on the virus, is a relationship between the virus and the red blood cells. So finally, I'd like to thank members of Ziping Yi's lab at SIBA who partake in the serology for the WHO selection process each year, and the team at VirusSite, Kevin Kohlmeyer, Mike Artiger, who helped get us set up with the virus counter and provided training. Thank you.
So one of the things that can affect these, whether you've got active particles versus inactive ones, is the time you run the cell culture. If I take the example of vaccinia production, as you culture it in cells, you get a linear, more and more virus particles come out. But as you go on longer, they become more and more inactive. So, I mean, I don't know whether you've sort of looked, I don't know, were these samples you got from commercial lots, or how long had they been cultured for?
No, they're not from commercial lots. So these viruses were all propagated in our hands. So after we got the initial samples from the CDC, we amplified them for a limited number, as few rounds of replication as possible. And, to try and keep everything on the same page, all the viruses were grown for the same amount of time.
But, I mean, we have approved cell-based flu vaccines. How do they look?
We don't measure the... so a lot of...
Because, I mean, you could have just produced them wrong. I mean, that's what I'm basically saying.
Yeah. So I do not measure the cell-based virus particles from a manufacturer. This is sort of part of our research mission, is to help us understand stuff so we can regulate it, and also help vaccine production. But the manufacturers themselves control what they do.
Yeah. Okay.
Vadim Sutninsky from PATH. Thanks for interesting presentation. I have a couple of questions. Does your data indicate to you that egg-based systems is somehow has a higher fidelity of making infectious particle than cell-based system? And second question, my understanding, a lot of clinical H3N2 samples cannot be isolated in eggs, maybe only about 5% are. So the viruses that you used for your studies, were they all originally isolated in eggs or in cells? Did you know if there's both systems that have been used to isolate original viruses?
So the first question, with regards to cell-based production or egg-based production, the infrastructure is there for egg-based production, and a lot of the reagents we produce are directed for egg-based production. What I was looking at was not so much the total amount of protein. I wasn't even looking at the amount of protein that could be obtained from either system. I was just looking at the number of viral particles. So if a system is already there and you can grow a lot of virus efficiently and you're happy with the production, I'm not advocating changing systems. What I was more interested in was when you start looking at the serological assays, the virus you're putting in is important, and how you quantify that virus is also important. With regard to the isolation of different viruses, I was limited to which viruses I could use based on which viruses had been, the same virus from the same patient that had been isolated in two different systems. And so when we isolate for the WHO serology, all the different groups that are isolating viruses, especially the CDC, they're isolating as many of the currently circulating viruses as they can, and that's what we want to use to see how the serological assays are performing. So we're going to use what's available, but it's just good to understand the differences.
Yeah, good talk, Bob Jiang, CDC. You showed the data with MDCK cells. Have you done work with Vir cells? Do you have any data?
No, I didn't do any virus amplification in any other cells, but that will be a good thing to look at. Thank you.
Yeah, I think it's very important to relate it to actual conditions that are actually used in manufacturing, because manufacturers are aware of these issues. I don't know if there's anybody here who works for one of these companies who can answer it, but I think you might be sort of damning cell-based systems here unnecessarily. I don't know the answer to that one. You don't seem to know it either.
I think this is not so much looking at the production-related issues. It's more of looking at, when you move into a clinical trial and you're trying to measure the efficacy of a vaccine, you're wanting to look at the antibody response.
Right.
And so the way you can measure the antibody response is, been done with the hemagglutination inhibition assay. And there have been difficulties with certain viruses. For some of the pandemic viruses, there was a move to use horse red blood cells because that's what they could get to work. So, but at the end of the day, you're still trying to look for the antibody titer. And so understanding that relationship between the antibody and the virus is important. And, this work was just sort of pointing out that, there are some limitations with some assays.
Sure.
And, some of the newer technologies are enabling us to see that, and some other technologies will enable these problems to be overcome.
Right. Okay. Thank you very much indeed. So let's move on to the next talk.